in Sunday. I'm very lucky to have this video made by our graphic department, Jim Stanley. So this just shows you a cartoon of the dynamic structure of the blood-brain barrier. It is consists of, many structures. For example, the inner layer is the endothelial cells with tight junctions, and then we have the parasites and the basement membranes, and then there's the astrocytes and the foot. So this is to protect the brain, the central nervous system from toxic materials, and maintain the equilibrium. For MI, there's common ways to measure BBB permeability and the function. One, of course, with gadolinium contrast agent. So under normal condition, the transendothelial permeability is very low. Only gadolinium can leak out with disrupted tight junction. Another way, recent years has been really growing interest is using water as an endogenous tracer. So water is a small molecule, and there's many routes for it to cross the brain-brain barrier, and it's linked to especially the ApoE, the AQP4 water channel function and the lymphatic system I will talk about later. So just to give a quick overview of the common ways, so dynamic susceptibility contrast perfusion weighted MI, very common, everybody doing it quite often, routinely, and from dynamic curve we can get hemodynamic parameters such as CBV, CBF. With acute leakage of the BBB, especially with disruption, you will see the delta R2 curve will have an undershoot and then causing underestimate of CBV. So there's many different ways to correct for this, but this is the common way proposed by Wiskoff and Jerry Boxman many years ago, is introduced this K2 and this parameter which indicates the permeability. So with this you can get both accurate measurement of perfusion as well as permeability. As shown in this study by Richard Lee, basically it shows in acute stroke, permeability increase was related to hemorrhagic transformation, and the higher permeability is more severe form of parenchymal hematoma. And another way to use BBB permeability imaging for detect subtle change in BBB permeability is using dynamic contrast enhanced DCE-MI. So the full model was characterized with the T1 weighted. The previous one was T2 star weighted with the TOFS extended model, but usually we ignore the backflux, and then it's the simplified PET-log quite commonly used. And this is a typical protocol we use at the USC with a 30 seconds baseline with a 30 seconds baseline followed by 16-minute dynamic scan, 30 seconds just to get a T1 map. Actually, this is a quite commonly used method, but it has, as you can see, shortcomings. So this is detect subtle changes. So K-trans is in range of 10 to the -3 to 10 to the 4th per minute. So it's really low. And it requires a long scan time. So, there's a baseline drift and also motion. Then we use PET-log model. Of course, there's because the injection of contrast, there's a lack of test-retest. So there has been a community effort. For example, this Harness group led by Joanna Waterlaw to harmonize and standardize the DCE protocol. So nevertheless, there have been success with the DCE-MI to detect subtle BBB change. So this is work published by Professor Beza Lukowicz group at the USC, just detecting, the hippocampal K-trans increase with aging and mild cognitive impairment. As a control, MS patient doesn't have this permeability increase in hippocampus. This actually 2 years ago they published in Nature showing this K-trans increase in hippocampus. It's increased with ApoE genotype as well as MCI. It's very interesting. It's not correlated with either A-beta or tau protein deposition status. For small vessel disease, there's evidence of increased BBB permeability in white matter, especially around the edge of the white matter hyperintensity. But the evidence hasn't been very consistent. For example, this group is Oxford group. They publish the increased KW in actually sporadic form of small vessel disease, but not in genetic form of small vessel disease, for example, Cadicel. And it doesn't coincide with this PET imaging of the microglia activation. On the contrary, other study, for example, this study by Dr. Uchida actually in Japan, he published a study in genetic form of small vessel disease, Cadicel, showing increased BBB permeability correlated with the QSM in genetic form of small vessel disease, Cadicel. There's also evidence of BBB permeability increase in Alzheimer's disease. This study was published by the Urich group in Netherlands. This shows increased permeability in AD patients. But if you look carefully, there is also a lot of white matter hyperintensity in AD patients compared to controls. So there could be a vascular factor. So just to summarize, so the DCE protocol, there's evidence of increased BBB permeability in aging MCI and cerebral small vessel disease, but the evidence is not very consistent. There's ongoing effort to standardize DCE protocols, such as by the Hannes group. So I'm spending the rest of the time talking about using water. So we talk about it is a growing interest. Why, first you don't need a contrast agent. And also water is a small molecule with 18 molecular weight compared to the GBCAs. And its place related to the aquaporin 4 function, which plays a key role in the lymphatic system. So this is a magnified view of the BBB. You can see actually there's many pathways for water to cross the BBB. It can be through passive diffusion as well as co-transport with the ion pumps. Then once you cross the endothelium cell, there's the aquaporin 4 water channel on the astrocyte endofoot, which is assisted diffusion. And there's the very hot theory about the lymphatic system with the brain drainage. So the theory hypothesizes the water transport from the arterial PVS through aquaporin 4 brushing through the parenchymal, then carry away the waste, and then flush out through the PVS of the venous system. And with aging, you can see the system break down, and then in Alzheimer's disease, then the system basically clotted with amyloid deposition and totally disrupted. So how do you map the BBB water exchange? There are many different ways, and our group has proposing this basically using diffusion weighting to differentiate the intravascular and tissue compartment of labeled water. For example, this is the curve of diffusion-weighted ASL signal as a function of p-value, and then we basically measure at a different post-labeling delay from short 1500 milliseconds to all the way to 2100 milliseconds. And you can see there is the fast decaying component, which relate to the vasculature, and the slow component relate to the tissue. So with post-labeling delay, you can see increase the tissue compartment indicating water exchange. And then you, through the basically by exponential model, then we have a theory called two compartment approximate passage model APM, and you can actually estimate KW, and we have replicated this phenomena in monkeys and also in rats independently by different groups. So this is the curve in the images in the human subjects. So this actually has been really challenging, just because the ASL signal is small, and when you add in diffusion, it's even smaller. So we have developed this diffusion prepared 3D grays. So we have a relatively stable image, although low resolution. This is in aged subjects repeated 6 weeks apart. We can achieve ICC of 0.75. With this technique, so we did a few applications. For example, this is collaborated with Dr. Brian Gold at the University of Kentucky. He basically applied our technique in a cohort of healthy aged subjects. But they have CSF A-beta measurement. So he found KW was positively correlated with CSF A-beta in the whole brain and also different regions. And because with higher KW is correlated with higher CSF A-beta, which means the parenchymal A-beta is lower. So higher KW means lower brain amyloid deposition, which is consistent with the lymphatic theory. And this is typical 3 subjects. So they have a CSF A-beta level of 188, 259, and 344. And then we can see the KW map correlates very well, while the CBF and TT is not. In addition, we can see the correlation with cognitive function, such as memory and executive function. Again, this is Dr. Ichita in Japan. He independently performed the study on Phillips. He did his own pulse sequence. So he found actually this cohort subjects that was carrying different one pair of ApoE4 as well as heterozygous ApoE4 and ApoE3, and one pair of ApoE3. So he found actually with higher load of ApoE4, KW decrease, QSM increase, and also PET A-beta 42 deposition increase. So this is correlated with each other as well as cognition. So this is independent validation. So the mechanism-wise, so this was studied by a group in UCL, led by Dr. Jack Wells. So they did 2 mice model. One is wild type, one is EQP4 knockout mice. And you can see, they use a slightly different technique of multi-TE ASL. So they measured the exchange time increased in EQP4 knockout mice. So exchange time is 1/KW, which means KW is decreased. As comparison, you see there's nothing changed with the ATT, CBF, as well as ADC. So there's the mechanism of AQP4, decreased AQP4 function with reduced KW. Recently, we also published 2 papers in a genetic form of small vessel disease cardiac, working with a group in Beijing. So you can see, with symptomatic from asymptomatic to control, KW increase. CVF also increase and ATT decrease. And this is another study we published.
Professor Wang, can you please
okay.
sum up soon?
Yeah, it's the same results. And so we have recently developed the high resolution version of this to be isotropic for 3 to 4 millimeter. Yeah. So this is our conclusion. So basically, it's feasible to do water exchange with non-contrast methods, and there's evidence of decreased KW with aging, with neurodegenerative and cerebral small vessel disease. Yeah, thank you, everyone. Thank you.
Terrific. Thank you very much for this comprehensive and honest assessment of the state of the art of brain-blood barrier permeability imaging. In the interest of time, we're going to move on to the next presentation,